So our next speaker, Dr. Emery Brown, is another one of these people I hold in awe because, like, I have one job and I find that that keeps me pretty busy. But he is a named professor of medical engineering and computational neuroscience at MIT. He is also a named professor of anesthesia at Harvard Medical School, and he's also a practicing anesthesiologist at MGH. So I'm not sure how you do it and also find time to come talk to us, but we're extraordinarily grateful. In addition to that, he is one of a very, very small number of people who has actually been elected to all 3 national academies, and in fact is the only anesthesiologist to have done so. So please join me in welcoming Dr. Emery Brown.
Great, thank you, Margot. Thanks for Zach and company for inviting me. I'm going to talk a little bit about anesthesia, and actually I'm having PTSD here because that last time I was in this room was for organic chemistry, so let's get through that, all right. So I'm going to talk about anesthesia today, So, and what I want to show you is that this is really a case study. And let me just be frank about something right up front. This is a high signal-to-noise problem. So I want to show you that what happens, how we should be looking at a high signal-to-noise problem. And actually, when it was treated as a black box problem, it actually turned people off and has actually had a tremendous clinical effect in having people not use an important information source. I want to illustrate that to you. All right. So here's what I'm going to talk about. I'm going to give you a little overview of what anesthesia looks like. We're going to talk about loss of consciousness induced by propofol and some of the other anesthetics. Just like Jennifer talked about aging, I'll show you the implications of aging. Then we're going to take a little bit of a look at the future. Like if we get some of these things down, where can really a detailed mathematical, statistical understanding of this problem help us as far as designing systems that could allow us to give anesthesia better, all right? So I'll tell you the punchlines right from the start. So under anesthesia, the brain is not turned off, right? Actually, what happens is actually quite dynamic, and it's the dynamics the alterations in these dynamics that actually induce the anesthetized state, right? And we think that this is one of the primary ways that the drugs actually work. It's not the only way, but it's probably one of the primary ways it works. So once the kind of the payoff here is that these dynamics are readily visible in the electroencephalogram. You can see them. And you can see the type of drug. You can actually see how the drug affects the dose of the drug, and you can also see the age of the patient. The response is a function of age. It's really very, just that obvious. You'll see it in just a second, all right? So that means you can use this to dose the drugs, and if it's as robust as I'm suggesting, you should be able to generate a control system. And then the other thing is if you get a sufficiently deep enough understanding of what's happening in the brain under anesthesia, as opposed to turning the brain off, maybe you can turn the brain back on. So I just want to hint at that at the end, all right? So actually, how many people here have had anesthesia? Oh, see, it's a great topic, see? Better than you realize, see? So what is anesthesia? So it's this drug-induced, reversible state. You're unconscious. You're not supposed to remember. It shouldn't hurt. It's nice if you're not moving around. And this is key right here. Drug-induced reversible, because without the reversible, all this is synonymous with death, all right? All right? So, and here's where we make our money as anesthesiologists, because we have stability and control of the physiological systems, all right? And so this is what's said, how anesthetic drugs create the anesthetic state is not known, all right? So we're going to address that in just a second. But let me just tell you how anesthesia is actually done. So the way it's carried out is we have different drugs which approximately produce the different states. So we would give a drug like propofol or one of the barbiturates or maybe one of the inhaled drugs to induce unconsciousness. We'd add drugs like the opioids. And part of the reason that we have the opioid epidemic now is we've used opioids almost exclusively to treat pain as opposed to using a multimodal technique. It's not the only reason, but it's certainly contributed to it. So they're the most effective drugs, but it makes sense that after a while, if you use them exclusively, they're either not going to work or people are going to become resistant to them, right? And that's indeed what has happened. So if someone is profoundly unconscious, you really don't have to worry about amnesia. But if someone's being sedated, you can give them a benzodiazepine, a Valium-like drug. And then we give you drugs which actually paralyze you, render you immobile. And if you look here, you see how I have in each category, I have the inhaled drugs. So we still use nitrous oxide at Mass General, but most places don't. But the inhaled drugs, the primary class of inhaled drugs are ethers. So we still use ethers in, even in 2018. So ether, as you may know, was the first anesthetic, the first public demonstration of it took place at Mass General Hospital back in 1846. We actually celebrate Ether Day at Mass General. We sit around and sniff a little ether. No. What we do is, it's your anniversary. It's called your Ether Day, but it sounds like something else, right? All right. So I'm going to show you sort of like what anesthesia looks like here. So this is, I call this a Zerothorder Experiment because it's a freebie. 60,000 people get anesthesia a day. So you can just see this very, very easily. So this is the EEG of a lady who is about 60 years old. She's going to have thyroid surgery. And I'm just going to give her a bolus dose of Propofol. Propofol was the anesthetic that was associated with Michael Jackson's demise. It's the drug that we use most commonly, all right, for induction of anesthesia. So I'm just going to play this little video here, all right? All right, so there are 4 leads of EEG, 2 here and 2 there. All right, so those are eye blinks. You blink your eyes, you see these large muscle artifacts. All right, now what's going to happen here in a second? What's going to happen in a second is that you're going to see like a lot of noise appear on the EEG. And it's because when you inject propofol into a small vein, like the one on the back of my hand here, it burns like crazy. All right, and for pedagogical purposes, that's actually going to be good. Right, so watch what happens. I mean, not good for her, but I mean, what I want to say to you guys, right? All right. So here it is. You'll see this noise. See this? She's tensing up like this because it burns. All right. And you see, it's getting large. You can see it across all the channels. Now watch what happens here in about 6 or 8 seconds. And I'll stop it here and show it to you. I'll stop it right there, all right? See, all that disappeared. You can see when the drug took over, right? So all that noise disappeared, and the dashed vertical lines there, the dashed vertical lines right here, is 1 second. So going up and down between here is about 12 to 16 times per second. This is what's called a beta oscillation. And this is the oscillation, like, if she were coming to me to have a colonoscopy, I would titrate the propofol to try to get her into this state. It's just a state of sedation. All right, now she's going to just go right through this. She's not going to stay there. It's just going to be for a second. Now watch what happens next. Boom, large slow oscillations, all right? So anytime you think slow oscillations, just like slow wave sleep, that's when the brainstem is most inactivated. The same thing is occurring here with her. In other words, her brainstem just became profoundly inactivated. And if you looked at her, she lost consciousness right at this point, all right? So you can see it. It's not subtle, all right? And then I'll just play it a little bit further. It's going to go flat and now burst. Now flat and burst. So this is a state called burst suppression, and you see it in hypothermia. It's a state of coma. It's a signature of coma. It means that we'll talk a little bit about the physiology in just a second. But here's what I just shown you. This was her awake, all right, like this. And here, the magnitudes of these are very important. So with her awake, her EEG is about 5 microvolts, all right? Then as she goes through these other states, so there's the sedative state where she was at about, with the beta oscillations, and these are the slow oscillations here, right? So these are about 20 to 50 microvolts. So she goes from 5 to 20 to 50 microvolts. And then down to here, the burst suppression, and if you look at the periods in between the suppression, she's actually flat, all right? And then this is the state. You didn't even see this state because she went through it so quickly. This is the one I'm going to talk the most about. It's called slow oscillation, like this, and it's called an alpha oscillation riding on top. It's about roughly 10 cycles per second, all right? It's slow oscillation and 10 cycles per second, all right? And this is the state that we would try to keep you in if we wanted to have you be anesthetized. Not too deep, not too light, but basically about here. And there's another state here, paradoxical excitation, which I'm not going to talk that much about. But why is this a high signal to noise problem? You can see it. See, as you get to deeper states of anesthesia, what the drugs are doing is they're making large parts of the brain work in concert, large numbers of circuits work in concert. And these are actually extracellular currents that are flowing in the brain that the drugs have actually hijacked, if you would. All right? So it's not surprising that you could use a very simple monitor. So if you just said, OK, I'll just measure when low-frequency power increases, you're going to have a rough idea, based on what I've shown you here, when someone is going to be anesthetized. All right? So that was one of the ideas that was behind. These are some of the commercial monitors that are available for tracking brain states of patients under anesthesia. GE has one, Masimo has one. The most popular is probably BIS. All right? And then there's one called Narcotrin, which is developed by a group in Germany. The way these monitors were built was they take EEG in real time, all right? And they essentially had a regression model that they fit to the data. And the regression model gave back a number between 0 and 100. And you were told that for this particular scale, if you keep the person between 40 and 60, that's all you have to know. You don't have to worry about anything else. We've done everything for you. Think not, because otherwise you'll get it wrong. So after a while, this doesn't work, right? So as a consequence, now people are quite skeptical of using the monitors because this was too simplified for them. This was about 20 years ago when this came into being. So I'm going to show you the advantages of taking the physiology and sort of the circuitry a little bit more seriously, all right? So the way I'm going to plot these is I'm going to show these graphs as spectra. So here is a young lady that I took care of a few years ago. She was having just a very small procedure, so we gave her propofol. And then here is the slow oscillations here in the beginning. You can see them. If you look at the spectrogram down here, this is time on this axis, frequencies are here, and the color tells you the amplitude of the oscillation. So the redder it is, the larger the amplitude of the oscillation. So she has, you can see in here now, two patterns. There's something at about 10 hertz, maybe about 15. That's the high frequency part of this. And then there's a part which looks like this, which is the slow oscillation. So this is her. This is the signature of propofol. When you give someone propofol, this is what you're going to see on the monitor. All right? So you started here, and you continue it. As long as you keep the drug running, that's the state she'll stay in. You turn it off, and she eventually comes out of it. All right? So let's look at some of the dynamics of propofol a little bit more closely. All right? So we've done a number of studies, getting people to volunteer, just recording data from the operating room, but we've also done a number of studies getting people to come in and volunteer and let us anesthetize them. And you're very safe. I mean, this is, you got 4 or 5 anesthesiologists there. I mean, like 4% of the anesthesia workforce at MGH, they're with one patient. I mean, so, it's pretty safe, all right? But this is one of the studies that we did. So we have whole head EEG, so 64 sites, all right? And we're going to do a very simple experiment. Just give increasing doses of propofol, all right, for about an hour, and then give decreasing doses. And watch how the EEG changes over time. So not only is there a temporal dynamic, I showed you that 10 hertz oscillation and slow oscillation, there's a spatial dynamic across the head, which is very rich, all right? And it's basically like this. So let me just set it up so you can really appreciate it. If I stand here now and I close my eyes like this, and I put electrodes in the back of my head, you'll see an oscillation, which is 10 hertz, in the back of my head. It's called the eyes closed alpha oscillation. Pretty much everybody has it. About 5 to 10% of people don't. It gets a little bit weaker as we get older. But it's very, very strong. And so that's what's happening here. So this is a young man subject. So the nose is up here, left ear is here, the right ear is here. And these are the 2 electrode sites right here, the left and the right occipital areas. And he has his eyes closed. So he has his eyes closed like this. And there's the 10 hertz oscillation. All right. Now what's going to happen as we give him increasing doses of propofol, this oscillation is going to leave the back of his head. It's going to move to the front. It's going to concentrate here in the front as we give him the propofol. We keep it running. And then when we turn the drug off, it's going to move back. All right? So there's a very rich spatial dynamic to this, too. I'll just play this video just to show you this. All right? So this is, no drug has been given yet. Now you administer it. Now watch how this starts to break up. See, it breaks up. So now it's appearing in the front. Okay? And this isn't even the highest level yet. You see, it's all gone back here. And now this is the highest level, 5. And now when we turn it off and let the drug wear off, so that takes about an hour to get up to 5 and about an hour to get back down, it disappears from the front and comes back. So it does this. And it does this, it stays there, and then it goes back. All right? So what's going on here? So there was something that Jennifer said which resonated with what I was thinking that I wanted to say here, and that is we have these behavioral effects. You lose consciousness, all right? We know what the molecular targets of the drugs are. Those have been known for years. But why should this behavior come about with the drugs binding at these specific targets? So that's where the EEG helps us out a lot. Because so one thing that we can do is we can put down where the targets are. So in this case, one of the ways that propofol works is it enhances inhibition. It binds to GABA receptors, and it makes the inhibitory interneurons more efficient. So they're everywhere. But one place where they're quite abundant is here in the cortex, and also down here in the brainstem, and also in the thalamus. All right? So this is something you could write down. You can draw this wiring diagram. This is the reality of what you see in the operating room. And this is what you see when you do a study. Now, I just have to tell you one other little factoid. That 10 hertz oscillation, when it appeared in the front of the head, it was highly coherent. In other words, if you calculate the global coherence or just the simple correlation coefficients at the alpha frequency, all the sites in the front of the head are moving in unison like this. They're quite tethered one to the other, right? So this is where the modeling comes in. So with the help of Nancy Coppell, who many of you know is a very well-known dynamical systems expert at Boston University, we worked out this model suggesting that what's going on here is that this phenomenon here is likely an oscillation going back between the thalamus and the cortex. So without going into all the details, the thalamus has certain properties which suggest that this is reasonable. The cortex can be driven down to this alpha oscillation, and then the two couple, and when they couple, you get this oscillation going back and forth at 10 hertz. So why would something like this make you unconscious? Well, here's where the size really matters. Remember I said the oscillation goes from being about 5 microvolts up to about 20 to 50 microvolts. So it's just like me talking to you, and you said, all right, Emery, what I want you to do is, I hear your lecture, but I want you to do it differently. I want you to give the lecture, and you get one frequency, okay? And you get to keep the amplitude like roughly constant, all right? And you have to tell me everything that you're saying, right? So what I'm saying now becomes wah. No, it's actually wah. It's actually very loud, right? Because the amplitude is much larger, and it's at a fixed frequency. So that's what's basically happening here between the thalamus and your cortex, we think, in this setting. And now we've done the experiments both in monkeys as well as in rodents, sticking electrodes in both places to actually show that that's the case. Now, what's wild about this is that that's the 10 hertz oscillation. The slow oscillation is doing something entirely different, right? The slow oscillation, the 10 hertz oscillation is totally coherent in the front of the head. The slow oscillation is like this, it's totally incoherent, right? And it's different from the 10 hertz, from the slow oscillation in sleep, right? And the reason I want to show you this is that this is actual data coming out of humans, right? So this is, Laura Lewis is one of our PhD students who finished up and was a junior fellow for the last 3 years and now is on the faculty at the Martinez Center in Charlestown. And so what she did for her thesis was conceptually very straightforward. Patients who have epilepsy surgery get electrodes implanted. They get electrodes implanted to discover the location of their seizure foci. Once the seizure foci are discovered, they come back to the operating room to have the electrodes removed and then to have a resection. So when these patients come in the second time, they have to have anesthesia, so you can actually record what the neurons in the human brain are doing when a person receives anesthesia. And that's basically what she did for her thesis. So here's this patient awake, and these are the neurons spiking. You see the neurons spike pretty much wherever they want to, and this is just some sort of very small, like low amplitude oscillation, all right, just like we saw when the woman was awake, right? And then here is what happens once the person's unconscious, and we're recording from this part of the brain here, so the temporal lobe, not up front here where you have the alpha oscillations also, but here where you just have the slow oscillations. And the colors correspond. And so look what happens here. You see this? This is this slow oscillation. When the slow oscillation comes on, the neurons can no longer spike wherever they want to. They can only spike in very limited locations, all right? So now, if the alpha oscillation didn't make you unconscious, right, because it's tying up the circuits in your thalamus and cortex, now across pretty much most of the cortex, the times at which the neurons can spike are limited into limited windows. And moreover, if they're not coordinated, when this location here wants to communicate with that one, it's going to be hard, maybe easier to do. But as soon as you move like a centimeter or so away, like over to try to communicate with this one's spiking at a different phase. So those are the reasons that you would become unconscious. Right? So what's the practical implication of this? Well, it just showed up 2 weeks ago, right? Now, so there's an article in the New Yorker called Awake and Under Anesthesia, right? So science writers don't want to write about anesthesia, to learn about, there's some marvelous things that those guys are doing, figuring out what's going on in the brain. They want to write about awareness, right? They want to write about, someone was anesthetized. In fact, they call me up, these journalists, and go, can you tell me about one of those experiences where you had a patient awake under anesthesia and you didn't know it? No, I don't want to talk about that. I don't look good in that way. And plus, it's not a public health menace, right? You would think that it was happening 5 or 6 times a week or something. It's not the case. It's like between 1 in 1,000 to 1 in 20,000 times. But it gets your attention, right? The practical implication is it doesn't have to happen in 2018. If anything that I've said here is even remotely correct, right? Just by monitoring the EEG, dialing someone down to these patterns, you can be certainly sure that they're not going to be conscious. The reason it's not as certain as that is that anesthesiologists don't use the EEG, right? That's the issue, all right? So the information is there to be used, it's just not used, right? So then there's some immediate corollaries, and the corollaries are that if you have a different anesthetic, because they have different mechanisms, they have different signatures, and they're readily visible on the spectra, just as I'm showing you here, all right? Different drug class, you change the drug class, you get a different signature because you generate different dynamics the same way. It's just like, think of it this way from an engineering standpoint. You have the brain, which has all these recurrent connections, 10 to the 10 neurons, and you have a number of ways that you can shrink it down to lower dimensional modes. And that's what these are. These are just lower dimensional modes, essentially, all right? And you can talk about the circuit dynamics that go along with any and all of these, all right? So what happens as you age? Well, so what I was showing you was someone over here. This is someone who's 30 years of age who's receiving propofol, all right? Here's someone who's 57, like I said, this guy could be this guy's dad, let's say, all right? And then look over here, 57, so then this could be like his mom, all right? So this is a lady, she was 81 years old. I took care of her. She had the tumor on the side of her chest that was the size of like an American football. It took the surgeons a better part of 6.5 hours to remove it. But you see, she has the alpha oscillations and the slow oscillations, but you see how they're much weaker, right? So as we get older, these oscillations get to be much, much weaker. And what we have to do is just change the scale on the monitor so you can actually see them. But one of the important implications of this is you can give the person far less anesthesia and achieve the same state. It's a fraction of the already age-adjusted dose that you're able to give them just by taking this into account. Here's the part that's a little sobering. These two guys are the same age. His EEG looks like hers. His looks like his, all right? And we age different physically. It's not surprising that our brains age differently. What's wild, I never would have thought about this, why would you see it under anesthesia? And it's because, think of it this way. Forget about anesthesia for the purposes of putting someone in a state so they can have surgery. Think of it as an experiment. You put in a stimulus. So the drug is a stimulus. And when you're young, here's the response. You generate oscillations like this. As you get older, your ability to respond becomes like this. And it has nothing to do with arguing for dementia or that sort of thing. If you just look at the anatomy of a neuron, you have the myelin sheath, which is the insulation. It breaks down. The cell volume declines. The dendrites are no longer as dynamic. You don't produce as much neurotransmitter. It's just aging, essentially. But the clinical implication is that you can take better care of patients by taking it into account. And this is just to say that we work hard over here to understand the properties of these things statistically. And then with Nancy's help, we've worked out mathematical model descriptions of all of these structures. So it's not just, pattern recognition. It's like taking it back to the biophysics, because the system is, as I said, a high signal-to-noise system. And here's just an illustration of that for burst suppression, right? Where this is burst suppression. So this state where you see the burst, suppression, burst, suppression. And this is the idea that there's energy modulation, without going into much more detail than that, all right? Here's the wild part. This is an experiment that we did, all right? So this is with the help of David Boaz and at the imaging center at MGH. And if you look here, the gray is the burst suppression. The red is actually recorded by NIRS. It's the oxyhemoglobin level, right? And the blue is the deoxyhemoglobin level. So this model predicted this, all right? So it shows you the benefits of actually obsessing about the details and trying to get the physiology right, because there's no way in the world I would have inferred this sort of structure here from just looking, in other words, just seeing the burst suppression here. But the modeling helped sew the statistics together, if you would. All right, so I want to say a little bit more about burst suppression. I'm going to talk about closed-loop control of medical coma. So you've probably heard about medical coma. In fact, it was in the news just about 2 days ago. You may have heard of this little boy who unfortunately died 2 days ago. His name was Rikus Roik in Florida. He was unfortunately bitten by a bat. All right? And he didn't get the rabies vaccine in time. And there is a physician in Wisconsin, Milwaukee, who had successfully treated a small fraction of people by putting them into medical coma. So you hear that and say, oh, the medical coma. What is medical coma? Well, you know what medical coma is now. Medical coma just means that you give propofol or some anesthetic until you get burst suppression on the EEG, and you just hold the person there. It's just a deep state of being anesthetized. And one of the reasons it's done when you have seizures is so that because you're decreasing the metabolic state, eventually the brain will flip out of the seizure state, all right? And hopefully you can then, treat them. All right, but these are some fairly well-known people who've been placed in medical coma when they had head injuries. Malala, Gabrielle Giffords, and also Michael Schumacher. So we had some rat volunteers come over to our laboratory and sort of test out this idea of putting them in medical coma. So what we do is we just read the EEG in real time, and you can see that the pattern it's a binary pattern. You're either bursting or you're not. You're bursting or you're not. So you can turn the EEG into basically ones and zeros. All right, so it's converted into ones and zeros, and so the ones are the suppressions and the zeros are the burst, right? And in real time, you can put down a target level, and if you think of the fraction of time you want to be suppressed as the target, then you can essentially build a controller that would go after this, all right? So let me just show you what this looks like. So here's an example. There may be an example. So right here, there's the target at 0.4, all right? This is the EEG. And this is the instantaneous rate at which the infusion pump is being changed in order to maintain this level. And the white line is what the controller is actually doing. So the target's supposed to be 0.4. Here's what the white line is getting. And so this is time in minutes here, all right? So now we change the level. We change the target up to about 0.8, right? And it's not doing too badly. And one of the things you can see is the dynamics that the pump has to adhere to in order to generate this is quite different from what it has to adhere to generate this. And now we'll raise it to another level, go up to about 0.9 or so, climbs up, and tracks reasonably well, all right? So why is this important? Well, actually here, just to show you. So you can do this. So I showed you this example here. This is going up, going up. If you go up and you want to come down, you don't have a drug that can turn the burst suppression off. You depend only on the kinetics of the drug. So you just have to let the drug wear off, and then when you get to the level you want to be, you just start your infusion again. So you can see it just turn the infusion pump off here. But it's pretty robust in terms of being able to control this reasonably well. So there's no approved closed-loop system for delivery of anesthesia at present. I mean, if the FDA controlled the auto industry, you'd never have a self-driving car. I can tell you that for sure, all right? So, and it is a real challenge to convince the FDA to let us, try something like this. We're on the verge of doing it, hopefully. But for medical coma, it's really important because in the ICU, when someone is in medical coma, they just set the pump, and then the nurse or physicians walk away, and they come back, hour and a half later and go, oh, we switch that. It's just like, imagine having a plane, right, where the pilot, sets the autopilot and then goes back and talks to the passengers for a half hour, right, and then comes back, right? I mean, that's what it would be like, right? So this is a perfect opportunity to try to, build a controller that could be used. So I'll tell you about one final thing. In anesthesia, we work on turning the brain off. The brains of a lot of people, particularly older patients, don't work that well after anesthesia, so we've looked at ways of turning the brain back on. And this is work which has been done primarily by my colleague Ken Solt at Mass General. And one of the reasons for this is to try to make sure cognitive function of patients is actually as it should be. It returns to normal. You can imagine if you're sitting there for 4 hours with oscillations going back and forth like this, that's just not natural. That's not going to be good for your brain. So again, once the rats finish the one experiment, they come over and volunteer for another one here. And so this is an anesthetized rat. All right, so they don't sleep like that, right? He's got an IV in his tail vein, and he's breathing in isoflurane. So he's totally anesthetized. All right, and so what Ken is going to do, he's going to inject methylphenidate. Methylphenidate is Ritalin, the same Ritalin that's used to treat ADHD. So he's going to inject it. He has to flush it in. So he pushes it in, and watch what happens. Now, we haven't turned the anesthesia off, right? Oh, he's totally fine. Don't worry. He just has to turn over. His feet are caught in the, right. He does make it. I've seen the end of the video here. All right. Go watch. Come on. He did it. All right, good. So, but what are we doing here? All right? So Ritalin works by activating this pathway right here. It's called the mesocortical, meaning midbrain cortical pathway. And it blocks the reuptake of dopamine. All right? It's an excitatory neurotransmitter. So as opposed to a drug that actually comes in and inhibits the activity of the anesthetic, it's doing something quite different. It's taking advantage of the fact that they're excitatory pathways, and it's driving an excitatory pathway. It's not like when you give naloxone. So when someone has an opioid overdose, you have the opioid that comes here like this, and then naloxone comes in and interferes and removes it. That's not what it's doing. This would be like it's coming in and driving this system to make it to excite it, right? So we're in the midst of a phase 2 clinical trial testing out this idea. But again, one of the reasons these ideas are so plausible is that we've been able to use the modeling and the statistics to actually sort of tie down these circuits in a more detailed way. And we've also done this optogenetically. right? So this is what I've told you. So as I said, anesthesia is not the state of the brain being turned off. It's this highly dynamic state in which, to the back, on the back, as a back of an envelope statement, you can say you put the brain in these sort of lower dimensional modes, and being in these modes is not compatible with being conscious or processing information in a normal way. I've shown you how you can monitor. I've shown you how you can use them for control strategies, and I've shown you how that you can maybe use them to turn the brain back on, right? And again, as I've said several times, this is because we have tight integration between doing experiments, statistical analyses, and mathematical modeling. So I think that one of the things that I think is very important as we start thinking about data science and large data problems is that trying to create situations where we can design experiments as opposed to just letting data flow into us, we know that it's going to increase the yield of information that we get out of it. And then one final point just about anesthesia. You see, this is the anesthesiologist's view of clinical neuroscience. You see how we sit in the middle here, and all these things sort of circle around us, So it's actually the case, right? So just take an example here, like pain. Like if you can give ketamine to treat pain, ketamine can also be used in low dose to treat depression, and it can also be used to simulate schizophrenia. Or like over here, as I mentioned, part of the problems that we're having with the opioid overdoses are us using too much opioids basically in the operating room. Maybe some of the ideas that I've shown you for bringing people out of anesthesia might be helpful for coma recovery. And we have drugs like methylhexatol, which at low dose actually induce seizures, and at higher doses can be used to treat seizures, all right? So there's this wealth of information that sits in anesthesia research that's not been exploited. And it's not been exploited because of the perspective that we have on our field. We view our field as a subfield of pharmacology and not a field of neuroscience. But as soon as you think of it as neuroscience, and then from my perspective, you take the signal processing seriously, you can unlock a lot of information. So I'll stop there. Thank you. Question?
Can you speak a little bit about anesthesia with epilepsy, where there's an uncontrolled seizure happening at one part of the brain? Mm-hmm. Can it be controlled from a different part of the brain when you put it into these rhythms?
Yeah, so the question is whether the relationship between using anesthesia to control control maybe an epileptic focus. So what I was showing you back here, I mean, I didn't emphasize it perhaps enough, but when people have uncontrolled seizures, if they come in and they're seizing, their medications have failed, the immediate therapy that they'll start in the emergency room and then transfer in the intensive care unit is giving anesthesia, giving general anesthesia, all right? So then the question is, can you stimulate in one area here and perhaps turn off a seizure, in some other area? There are attempts to do this, all right? And it depends critically on the nature of the seizure and the area and what's connected to it, right? A lot of the seizures, one of the common areas where they occur is here in the medial temporal lobe, in this area here. And there are some connections perhaps coming from maybe the central part of the thalamus, which could be activated to do that sort of thing. So this is a very, very active area of research in a number of centers, both here at Mass General, like at UCSF, and a couple of other places. So I think the jury is out. No one has a robust way of doing it at the moment. But arguably, as we get some of this physiology down, it seems quite tractable.
So you've been able to show a few different drugs and how they interact with them. Mm-hmm. I assume that anything that affects the brain itself, any drug that actually has some effect on the brain, you can essentially see out of EEG.
pretty much most drugs affect the EEG. Yeah, certainly the anesthetics. I guess kind of the take-home message here is that these effects have always been there. And we knew they were there to some extent because people could build monitors, commercial monitors. Or let me say it this way. If you go back to 1990, there was no automatic device that could read and classify sleep. But in 1990, they built devices that could tell you approximately the state you were in under anesthesia. So it had to be a high signal-to-noise problem. And then what happened is that they just were content with saying, oh, when the power, essentially in the low frequencies goes up, you're anesthetized. When it goes down, you're not. I mean, that's a bit of oversimplification, but that's not too far from wrong. And then what I've just shown you is that the dynamics are highly systematic. Drug class, drug dose, age. So you have essentially 3 robust covariates which you can use to then understand the state that the person is in. And I think that's where I see a tremendous amount of promise, because you can process this in real time and then give physicians, anesthesiologists, very precise information about how they should dose their drugs. So I'd say this for the anesthetic drugs. It's not necessarily true for other drugs that you see this degree of dynamics. Question here? Oh, sorry.
There was one plot you showed in which you were showing the age-related differences.
Mm-hmm.
Is that difference also seen in the coherence patterns as the wave goes from back to front?
Yeah, there are differences in the coherence, and in fact, what happens is, as you age, I didn't put the plot in here, the coherence gets weaker and weaker over time. It's kind of like almost what you'd probably predict. Yeah, it gets weaker.
Okay, let's thank our speaker.
Yeah, thank you.